for DNA. Phosphate to the 3' position of sugar and to the 5' position. So if we go back just for a moment and look at this little cartoon, where we can see we've got several parts of the rung of our ladder. So here's the phosphate bound to this part of the sugar. Well, this phosphate is bound to that part of the sugar. This phosphate is also bound to that part. And we've named these parts of a sugar. We've called them the 3' and 5' ends. So that way you know how the sugar is oriented, and it's going to help us in terms of figuring out how DNA makes copies of itself. Well, what in the heck is a 3' 5' position? You start right here. That's number 1. That's number 2. That's number 3. Number 4. And number 5. Right? 1, 2, 3, 4, 5. So if I want to add a phosphate, I have to stick it right here. I can't put it here, I can't put it there, because that one's dedicated for our nitrogenous base. So it's always 3' to 5'. Another sugar would come up here. Then another sugar, that would be its 3'. There's its 5'. The next phosphate would go up here. And we just keep repeating the kind of zigzag pattern along the length.
So the carbons are what are labeled on the sugar?
Well, not really. I mean, you can't look at them. We number them, yes.
Right, but I'm
Yeah, so you start here, that's number 1, number 2, number 3, number 4, number 5.
So can you only bond phosphate to 3 and 5 and the number 3
Yep,
and
that's
number
it.
5
It
position?
won't fit anywhere else.
Okay.
All right, so why is that so important? Well, here's RNA. Now, this is what's different. Remember that we have 2 oxygens on our sugar. See how it looks like the home base of a baseball game, home plate? Right, that's the shape of ribose. But if we look over here, since it has both oxygens, we don't call it a deoxyribose. We can just call it a ribose. The other thing that's different is that in the RNA world, there is no thiamine in terms of a nitrogenous base. RNA does not use thiamine. It uses adenine, it uses guanine, and it uses cytosine. But it had to be different. So it uses something called uracil. So if you ever see uracil in the code, you know you're looking at RNA. Because DNA does not use uracil. And if you see thiamine in the code, then you know you're looking at DNA and not RNA. Because RNA doesn't use thiamine, it uses uracil.
So is uracil kind of be where thiamine's at?
Yep.
Practice with that in?
Exact same place. So that means really there's a total of 5 different nitrogenous bases. All right? A's, C's, T's, and G's, and U's. But now they're specific. DNA doesn't use the U's, and RNA doesn't use the T's.
So for the uracil, it doesn't belong to the category of purines and pyrimidine, right?
Well, look at its shape.
Is that
So
is that
which one does it look like?
Pyrimidines?
Yep.
So it belongs to the category of pyrimidines.
So in a way, it's just a different version of this one.
I see.
Mm-hmm.
On the previous slide, for slide number 8, it has the deoxyribose sugar molecule, but then for RNA, it says use the sugar ribose, not the deoxyribose, but they're the same shape. How do you tell the difference?
So look at the very bottom. Look at the name on slide 8 where it says the actual word sugar, right? See how right here all you have is a hydrogen?
Yeah.
There's no oxygen there, right?
Okay.
Now look at the next slide. That position has an oxygen there.
Okay.
That's the only difference. And in nature, that's a big difference.
So the OH is an oxygen?
Well, the OH is a hydroxyl. It's also
Right.
called an alcohol part. So sugars are actually alcohols.
Okay. So the deoxyribose doesn't have that?
Nope. See how that number 2 carbon only has a hydrogen bound to it.
Right.
Number 2 carbon over here has a hydroxyl bound to it instead. And that's a big difference in organic chemistry. All right, so here's another way of looking at the rungs of our ladder. Right? We can see the alternating pattern for the backbone of sugar, phosphate, sugar, phosphate. So we could go 1, 2, 3 prime, 4 prime, 5 prime, 1, 2, 3 prime, 4, 5 prime, 1, 2, 3 prime, 4, 5 prime. So it's 3 to 5, 3 to 5, 3 to 5, all the way up. And the same thing happens on the opposite side. Now the nice thing is, once you remember and get comfortable with the idea that they're like married couples, right? A's only go with T's, and G's only go with C's. That if you know one side of the ladder, then you can build the other side. Because you know you have to put a T here. And you know the backbone, it's just sugar, phosphate, sugar, phosphate. We know that if there's a C here, you have to have a G over here. And this is what enzymes are going to do. It's just laws of chemistry. If there's this there, I can only put that there. I can't put these two in this position.
You can put a U there.
If I have U's.
This is a DNA.
But if this is DNA, then my supply store, my pantry, my kitchen doesn't have any U's available.
The RNA.
Don't make life complicated, Mohammed. My God. Okay, so let's start making some copies of DNA. So what we've done is we've taken our spiral, or so known as the helical shape of DNA, and we've straightened it out like a ladder. Now, if I want to make a copy of it, and then John just said, well, if you know one side of the ladder, then you can automatically build the other side. Well, right now I don't have any access to either side because they're bound together, like my hands. I need to separate them. Well, remember we talked about how strong hydrogen bonds are? How strong are hydrogen bonds compared to maybe covalent or ionic? They're weak. That's kind of a handy thing. If these were covalently bonded, we'd probably be screwed. We wouldn't exist. So, I need something that helps break bonds. What helps to break bonds?
Hydrogen.
Yeah, something else. No, what kind of protein? What kind of protein are you thinking about?
Something made up of.
But like what?
Water breaks shit.
So what are the functions of proteins? Name me some functions of proteins. Enzymes. Are enzymes proteins?
Yeah.
Are all proteins enzymes?
No.
No.
And do enzymes help to either make bonds or break bonds? Oh. What kind of an enzyme do I need? I need helicase. Remember, it ends in ASE, it's an enzyme. And helical for the spiral part, right? So it's kind of a neatly named enzyme. This will help us to break the hydrogen bonds between the nitrogenous bases of your DNA molecule. So now we have them over here, right? They're separated. Well, we call the two blue parts the parental strands, like a parent. And in one way, this is definitely, you can tell it was a male-dominated world. The gray parts, we're going to call them the daughter strands. The daughter strands. Like if I had a girl offspring, she'd be my daughter. The
So that
grays.
means it's weak.
Okay.
That means it's weak.
All right, so helicase enzyme is going to help break all the hydrogen bonds. Now I've got just this blue part. Imagine the grays are not there anymore for the moment. I've got this side exposed. I've got this side with these parts exposed. Well, now I need to make another bond, right? I need to now kind of fill in the missing pieces. Another enzyme called DNA polymerase will join the nitrogenous bases and also build those sugar phosphate backbones. So the DNA polymerase will build the opposite side, or what's called the complement side. It'll do it to this side, and it'll do it to this side. And since it's laws of chemistry, where A's only binds to T's and G's only bind with C's, DNA polymerase really can't screw up that much. It's simply just putting, a 2-hydrogen bond to a 2-hydrogen bond, a 3-hydrogen bond to a 3-hydrogen bond. by the time we're done, we have a copy of our DNA. Because if we look at the pattern, right? We have AT, CG, AT, AT, CG, CG. And these match exactly. If we think about these guys, right? These are nitrogenous bases. Well, there's a pair of nitrogenous bases. Another pair of nitrogenous bases. Another pair of nitrogenous bases. If I read this ACGAT, right, it's a sequence of letters, right? So it's called a base pair sequence. I now know the sequence of the base pairs. Even if I only had one side, I would still know the base pair sequence because I know who could only bind to A's, would be a T. So even though I only see half of it, I can still tell you what the base pair sequence would be. All right, so it's language. We know these are nitrogenous bases, right? So the sugars and phosphates would be right here. The sugar's there, that'd be a phosphate. There's a pair of them, so it's a nitrogenous base pair. And we just simplify and call them base pairs. And there's a sequence of them, so it's a base pair sequence. So when someone reads your DNA, they're reading the base pair sequence. They're reading the ACs, Gs, As, Ts, and on and on and on and on and on. And these are hundreds of thousands of base pairs in length, just for one part of one chromosome. So we have trillions of these inside of us.
Do they always go in that exact order, A, C, G, H?
Oh, heck no, no. So here's the really cool thing. In DNA, we only have 4 nitrogenous bases, right? A, C's, T's, and G's. In the English alphabet, how many letters do we have?
20.
We have 26. I only have 4 letters. So now, picture a page in a book. Combination of letters. All right? I have a T, I have an H, and I have an E. That's the code for the, right? I have an A, and a T, and a C. That's the code for a cat. If I change just one letter, I can turn a cat into a hat. Or I can turn a hat into a bat. And then use the bat on the cat, who's wearing my hat. All right? But that's the cool thing is that if you look at every single person here, right? No two of you are alike. We now are actually changing the way we think about an identical twin. They're not identical. There are subtle differences. That means that we're only using 4 letters in combinations that make everybody unique and different. It's not a very big alphabet, but it's making trillions of different people. We're going to kind of see the advantage of that in a moment. All right, so how does DNA make a copy of our strands? So the order or the direction that DNA is replicated, that means a copy being made. I want to replicate the strand. DNA polymerase works in a 5' to 3' direction, adding new nucleotides only at the 3' end of a deoxyribose sugar. First, think of this analogy. Trey owns a sailboat and he wants to go sailing over an ocean shores. He's not going to sail to get over there. He's going to hook it up to a pickup truck. So you've got a trailer for your boat. Does the trailer for your boat go onto the front part or back part of your truck? It goes to the back part, right? Right? Because why? There's a hitch there. That's the 3' end of his truck. The front part where the engine is the 5' part of his truck. And so we have polarity in a sense. There's always going to be a 5' end and a 3' end, no matter where we are. Right? There's my 3' sugar. If I remove this whole piece right here, just get rid of it, well, there's still the 3' end. Get rid of all of this. There's still the 3' end. So if I want to build this side of my ladder, do I add it up here or do I add it down here?
Down.
I add it down here because there's the 3' end of my exposed ladder. It's always the exposed end. So I add another nucleotide to it. Then I'll have this, but now it's down here. So DNA polymerase comes along here, and now let's say, here's my hand of polymerase. I've made this, I'm right here. I want to make another sugar phosphate bond, and then add another nitrogenous base to it. So I'm going to stick a phosphate or a sugar here. Stick a phosphate there, then stick a sugar there, and then add my nitrogenous base. Well, if my thumb is the 3' end and my little finger is the 5' end, I stick a phosphate here, put a sugar here, there's my 3' end for the next phosphate. And so when I go down this way, DNA polymerase, remember, we only have half of a ladder really. I'm walking down this way and building. If I want to build this one, DNA polymerase has to go in this direction, which is why I put the big orange arrows in there for you. Remember, when they're separated, the DNA polymerase will go from 5 to 3, and if that's the rule, well, my 5 is now down here, and my 3's up there. So on this side, the polymerase walks in this direction, always adding to the 3' end. Always adding to the 3' end. So it's going to grow like this. Remember, they're separated. They're not hooked up together. They're two separate strands.
So how do you know which nitrogenous base to add after you add your phosphate and your sugar?
Well, remember, I'm reading off of a copy, so I'm trying to build this piece, is what I'm actually doing.
Okay.
But then, if I'm going to be using this piece to build that piece, I'm going to walk in this direction. Remember, they're separate, so this doesn't really exist. So if I'm DNA polymerase, then I'm going to build this. So I'm going to come down here and go down in this direction. It takes a little while to get used to.
Okay.
So hang on.
Okay.
All right, so let's look at what a gene is. First, back up to what a chromosome is. It's one long, continuous, uninterrupted DNA molecule. So now let's unravel it. So here's my unraveled, continuous DNA molecule. I then realize that there are certain regions that have information to make something. And we call that region a gene. A gene is the recipe or the code for a protein. It tells your cells how to make that protein. So genes code for proteins, and that they are a region, a specific region of a DNA strand.
So are they related to molecules?
Well, well, what's a molecule?
Long strand of DNA unraveled.
Well, no, what's a molecule?
Oh, a molecule. It's a mixture of atoms.
Are these atoms?
Yeah.
Whole crap load of them, aren't they?
Uh-huh.
So DNA is a big honking molecule made of lots of atoms, and a gene is just a certain region of that DNA. Think of it in terms of this. All right? Imagine a movie, the movie film, the literal film. And we've got different scenes of the movie. Right? Scene 1, scene 2, and scene 3. Well, let's make the scenes the gene instead. It's just a part of that one long strand. And in that strand is information to make a certain protein. But in that part is information to make a different protein. Does that help?
Yeah.
Let me try one more time.
Okay.
Take a cookbook and lay all the pages. All right, it's one book, right? But then I have a chapter on pastry, a chapter on pork, a chapter on beef. Those chapters are genes. The information on how to make that dish is in that chapter. The information on how to make that protein, a protein, is in what we call a gene. So there's the information. But all that really is a certain region of my DNA molecule.
A genius.
Uh-huh. So then what I really know is that this is really a nitrogenous base or a base pair sequence.
How do we know what protein they're making?
Well, we had to figure that out. What you do is you take the gene and make it do something, then you realize what the protein is, or you break it so it doesn't work and see what doesn't show up. And it's taken us years to do this. So you'll get a better understanding once we understand really how we make DNA and more the use of what a gene is. Okay? Our school is a gene on the DNA of Broadway Avenue. We're just one part of that big thing. Okay? Well, why are we looking at this? Well, so here's a chromosome. It's also, remember, one long DNA molecule. Other DNA molecules. There are patterns we have noticed over the years in trying to figure out what kind of a protein that gene makes. And we started to notice consistencies, patterns in everybody's DNA. And we call these patterns STRs. Short tandem repeats. Everybody has them. Well, these are the actual names. Don't memorize them. But right there is a repeat called the TPOX. And here's one called the D351358. That's really exciting. Okay? But what is it? Well, let's go back to our DNA molecule just for a quick sec. Let's go back to here. Right? This is a strand of DNA, right? It's also a part of a gene. And this is the sequence of bases, right? Well, what if now I took my sequence of bases, I took my DNA, and I'll lay it out lengthwise this way. And we're going to have really A's, T's, C's, and G's, right? We're just not showing you the other half. So we'll have a T up here, an A here, a G here, and a C here. Well, what if you were reading my genes, reading my DNA, A, T, C, G, C, G, A, T, C, G, and you went, this guy stutters. Wow, look at that. That is the same as that, and that's the same as that. The pattern is repeating itself, right? Well, everybody has these, not actual letters, in their genes. But the question really is, what combination of repeating parts of your DNA do you have? The odds of, let's say, you and I sharing one repeating segment is not bad. It's about 20%. But the odds of me sharing two parts of my genes that have the same repeating pattern are much less. The odds of you and I having 3, 4, 5, 6, up to 15 matching repeating patterns is basically incalculable. So when you guys watch TV shows like NCIS, CSI, any of these crime shows, and I want to know, well, I'm always picking on Trey. Pick on Sam for now. I think he's the one that broke into my house and killed my cat. So I'm going to, you ever heard TV shows say, well, how many hits in CODIS did you get? CODIS is how we have made copies, and we have made a library of everybody's repeating patterns once I have access to your DNA. Well, how are they using CODIS for these repeating patterns? If they are simply a repeating segment of your genes, then when you look at them laying out, so here, for example, we've taken their genes, right, and we're just going to make color codes. See how this pattern matches that one, and that pattern matches this one? So this pattern repeats 6 times for this person on their DNA. Now they got this is from their mom. That's their mom's DNA that's now in their cells. Remember, you get one from mom, one from dad. Well, the dad's version, they have the same repeat, but it doesn't happen as many times. It only happens 4 times. And I can use that to differentiate mom from dad. This person down here, there's their mom, there's their dad's, and they have the exact same pattern, right? And the same number of patterns. This one repeats 6 times each. That's a 6 repeat, but only a 4 repeat. These are 2 different people. Come down here, this person only has the repeat that only happens twice. And this person, it happens 8 times. And I can use that to identify people.
So do twins have more repeating patterns?
Do who?
Twins?
Twins would, yes. But there's still a variation where you might have gotten all of them. So maybe instead of 14 or 15, you might have 12 or 13 perhaps.
But is that what like, go ahead, sorry.
What does it mean when one strand repeats 6 times and versus the other one that repeats 4?
It just means that they're different strands.
Okay.
That's all that it means.
But is it only in identical twins that have the repeating pattern?
Now, everybody has repeating patterns, right? We share repeating patterns. But the odds are saying that, what are the odds of you and I having, we know there's 15 different ones in our genes overall. We've found them so far. What are the odds of you and I having exactly the same numbers of repeats in all 15 portions of our genes? So let's say I have 6 copies, you have 6 copies for every single different type of repeating region. It's 1 in several quintillion.
But isn't like a characteristic of like having brown eyes be considered like a?
That's a totally different thing. That's a whole gene.
Oh.
We're only looking at part of a gene.
Oh.
Like in that cookbook idea
Yeah.
on how to make pork, we're only looking at the repeating use of barbecue sauce.
Okay.
It's only one part of the whole recipe.
Oh, okay.
So now the problem is this. How do I know what your repeating segments are? Well, I need your DNA. Well, how do I get that, right? They take their little cotton swab, right, and they swab the knife, right? Or they swab the doorknob. Or they get your epithelial cells under your fingernails. Well, all right, so that's the bad dude's DNA is in the fingernail skin. But I need to know if this skin I found here actually matches John's DNA. So you got to go to John and take a cheek swab of me and see if the DNA in my mouth matches the DNA found under her fingernails. And if they match, then it's probably my skin. I'm probably the one that attacked her. Well, how do you know that we're the same? Because we're checking the number of repeats. We're looking for the STRs. And if they match, we're the same person. That means I'm the guilty individual. Well, the problem is that when you get DNA in the cell, there's not very many of them. You can't read a single strand. There's no way we can do that yet. I need to make lots and 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 lots of copies. I have to amplify the signal in a sense. So how do I make lots and lots of copies of someone's DNA? We use a process called PCR. And I want to show you a video right now, if we're on a time, of how we do that before we go anywhere else. So let's look at how we make a copy of DNA. And I'll post these on Canvas so you guys can watch them again over the weekend if you're a little bit lost in terms of what the heck are they doing here? Because we are kind of towards the end here.